Welcome to today's lecture. In this class, we're going to be talking about what flow cytometry is and what you can do with it. So to give a base definition, cytometry is the measurement of the physical or chemical characteristics of cells or other biological particles, looking at them at a single cell level. And it's important to note that this is not limited to flow cytometry. So as a cytometrist, you really are a cellular detective. Your job is to establish a cellular identity. And to do this, you need the power of resolution. So you want to look at things on how to determine cell type A from cell type B and distinguishing their separate identities. But this can also be determining what your cells are doing. So part of your interrogation 101, the things you generally look at are: what do your cells contain, express, or produce? What do they do? So what is their function? What do they look like? And who do they associate with? Some standard techniques for looking at these characteristics of cells include flow cytometry, microscopy, and genomic cytometry. And I'm sure by now in your degree you've heard of the first two, but maybe not the third one. So flow cytometry gives you zero spatial resolution. What I mean by this is you can see if your cell expresses a marker or not, but you can't tell where in the cell that marker is expressed. Now, general flow cytometers that you'll find most places can measure about 20 parameters, but we have an asterisk here because this is changing rapidly. Now, this technique is highly quantitative, so you can look at a large number of cells and a lot of things on the individual cells, and you can look at millions of cells in a single sample. So flow cytometry is a fast, sensitive, and quantitative multispectral analysis of a large population of cells. Microscopy, on the other hand, has really good spatial resolution. So you can see your cells and you can see where in the cells whatever you're looking at is expressed. However, generally you look at many fewer parameters, so usually 1 to 6. It is also much more time consuming, so it can be less quantitative because generally you look at fewer cells. So you can think of microscopy as a small cell population, low throughput, but excellent spatial resolution way of looking at your cells. Now, genomic cytometry is looking at the genomic contents of your cells, but in a single cell fashion, so not on a population. So it is super high dimensional single cell profiling, where you look at hundreds to thousands of genes per cell, and you look at these in tens of thousands of cells. Now, there are potential issues with doublets, and gene dropout. However, this is a really powerful technique to look at genomic expression on a single cell level. Now, today we're mostly focused on flow cytometry, so let's dive into this a little bit more. So what is flow cytometry? Well, breaking down the name, metry is the measurement, cyto, of cells, flow in a fluid flow. So flow cytometry is the measurement of cells in fluid flow and is a technology that allows analysis of multiple characteristics of particles or cells as they flow through a beam of light. When we look at flow cytometry, we're generally measuring or looking to measure single cells, and we're looking at the light that we get from these cells. So this can either be the scatter properties or the fluorescent signatures, and we'll talk more about this in a little bit. So if we think about this in terms of candy and resolution, flow cytometry is kind of like measuring fluorescent gumballs. So we can see what color the gumballs are, but we really don't get any other detail than that. Microscopy, on the other hand, we can see where those colors are. We can see all the various features of the cell. And so that's really one of the main differences between cytometry, flow cytometry, and microscopy. Now, no matter what flow cytometer you deal with, you're going to have the same basic components. So first up, we have the fluidics. This is a stream of fluid that transports your particles or your cells from your sample through the machine. So you can see this happening right here, where we have our particles coming into the machine and flowing through where they're interrogated by the lasers. Next up, we have the optics. That's the series of lasers and filters that allow you to detect your fluorescent signature on your cells. So the lasers intersect with your cells at the flow cell, so that's where we excite the fluorochromes. And then the light from this is directed to a series of optical filters where we detect the emission of those fluorochromes. And finally, we have the detectors or electronics. So these are downstream of the filters, And these are the parts of the machine that convert the light signals into electronic information that can be processed by a computer. Now, in our facility at the Faculty of Medicine and Dentistry here, we have three main kinds of flow cytometers. So the first up are your analyzers. These are the standard units. You get your forward scatter, your side scatter, and your fluorescence, and this is what is generally used to analyze cells. Next up, we have the sorters. So in addition to all the parts that an analyzer has, these have the ability to recognize and sort populations of cells. So they take a heterogeneous population of cells to start with, and then can sort them into very distinct, homogeneous populations of a certain type of cell. So when you hear people say FACS, this is actually sorting. FACS stands for fluorescence activated cell sorting. So it's not an acronym for all of flow cytometry, merely just for cell sorting. And finally, we have the imaging cytometers. So this is similar to an analyzer. However, for each cell that goes through the machine, you also get a fluorescent image of that cell. So it's kind of like a nice hybrid between a flow cytometer and a microscope, where you get the benefits of throughput from a cytometer, but the benefits of resolution from a microscope on a single platform. So it's a very powerful way of looking at cells. So let's move this out of the way. Why do we use flow cytometry? First up, the most important thing is we're analyzing a distribution of cells. So we're not looking at an average. So I'm sure you've heard of or maybe even done Western blots before. And you know in this technique, you're looking at an average expression in a population. By that, I mean you've taken your cells, you've lysed them, and you've taken that entire lysate, which is the pooled contents of all of your cells, so an average, and looked at that expression on a Western blot. However, by averaging everything, you can mask differences in that population. So if you imagine a population where you have a small percentage of cells expressing a large amount of protein, this could look like a specified intensity of band on a Western blot. However, on a flow cytometer, we can see we have a large negative population and a small, very bright positive population. Now, if we imagine another scenario where we have a population of cells where about half of them express less protein and the other half express none. Again, our Western blot, because it's an average, looks the same. However, in flow cytometry, you can see we have a small negative population and then about an equal, but not as bright positive. And same thing for this scenario where we have a larger positive population expressing less protein, same Western blot, but a very still distinct positive population. And finally, the scenario where all the cells express a low amount of protein, we can see that still on both platforms. So by analyzing this single cell on a single cell level, we can better understand what is happening in the entire population. It also can analyze many thousands of cells quite quickly. Like normally, most cytometers can run on the order of 5,000 plus cells per second. So you can get through a sample in a really robust and quick manner. And because of this, you also have very robust statistical information for looking at your cell populations. So some common flow cytometry assays, because of how the technology works, really anything you can turn into a single cell, you can study on a flow cytometer. Now, it has long been thought of as a technique for immunologists, and it still very much is. However, there are a lot of common applications. So things like looking at cell cycle or viability, activation of signaling pathways, drug delivery, activation, differentiation, phenotype and identification. The list is really unlimited. Well, it is, I guess, eventually limited, but there is a huge number of things you can study on a flow cytometer. The list really goes on and on and on, and depends on how creative you want to get with your samples. So let's loop back to the fluidics, optics, and electronics and dig into each of these a little bit further. So you'll remember the fluidic system. The purpose of this system is to transport your particles in a fluid stream past the laser. This is the flow part of the flow cytometer. Now, there are two important considerations. First up, the stream should be positioned so that it's centered in the laser beam. So essentially like a microscope, our cells need to be in focus to properly excite the fluorochromes. Because we want to look at single cells, we also need to make sure only one particle is passing through the laser at a single time. So this is accomplished by something called hydrodynamic focusing. So if you look in the zoom in of a flow cell here, you can see we have our sample right here. And then on either side, we have something called sheath fluid. So this is the fluid that transports your cells through the machine. Now this sheath fluid is flowing at a faster rate than your sample. So because it's flowing more quickly, it forces your sample into this funnel, which is what develops that single file run of your cells through the flow cell. And so this drag effect narrows the core into single file particles. Now it's really important to note that this sheath fluid and your sample core don't mix, they stay separate, and that is again because of this hydrodynamic focusing. So again, we have this sheath fluid that is flowing faster than your sample. This forces your cells into this single file line through the flow cell and also keeps your sample running through the machine. And this hydrodynamic focusing is based on the principles of laminar flow. So on a flow cytometer, we have different flow rates. So we can run a machine on, say, low, medium, or high, or sometimes you can choose the exact speed. So what happens here is when we change the flow rate, we change the amount of pressure on the sample. So how much sample we're pushing through a machine at once. So as we push a sample through a machine more quickly, so over on this side, you can see our core stream has widened. So it is exerting more pressure onto the sheath fluid. And so this runs our sample through the machine more quickly, but also increases the chance that more than one cell will pass through the flow cell at any given time. You can also have cells that pass through out of focus for the laser beam. So if we imagine that this is the plane of focus right here, as we increase our speed from low to medium to high, we're increasing the pressure that the sample is exerting on the sheet, so we get a bigger core stream. We have a chance of more cells going through at once, so coincidence, but also a chance of more cells going through the machine out of focus, which diminishes the resolution. So the fluidics play a really important role in getting good, clean data out of your samples. The slower you run a machine, the better resolution you'll have to detect your fluorochromes, but also you'll have a better population of single cells. You won't have as many multiple cells going through together, so you will have a better understanding of your population of interest. So just to go over this one more time, the fluidic system works on a principle of hydrodynamic focusing. So the sheath fluid runs alongside your sample. It's at a faster speed, so this creates a drag effect, which brings your sample through the machine, but also creates that narrowing effect on your core stream so that your cells ideally go through one cell at a time. Now, the width of this core can be adjusted by increasing the sample pressure. So the faster you run a sample, the more pressure that is exerted on your sample, the wider this core stream gets. Again, this will increase the risk of running more than one cell at a time through a laser or running cells out of the resolution plane of the laser. Now, to function properly, this sheath fluid needs to be free of debris, free of bubbles, and properly pressurized. So that's how you get a nice clean set of data, is by having really great fluidics. And finally, we have our interaction with the optic system that occurs in the flow cell after focusing. So in this area right here, after our cells have been nicely focused into a single file line, that's when we hit them with the lasers. So the optic system is a series of lasers and detectors that provide the information about a particle's properties. In this case, we're talking about light scattering and fluorescence emission. So light scatter, you've probably heard of this before. We'll move this out of the way again. This is where you get the information on the relative size and the relative internal complexity. It's probably what you've heard it called before. So forward scatter, or FSC, is the size. Now I put this in quotes because it's not truly a measurement of size. It's actually a measurement of diffracted light. So you can imagine your forward scatter detector is kind of like a wall capturing a shadow. So if we have an object sitting in front of a high-powered light, you can imagine a small object is going to create a small shadow on that wall. A large object is going to create a large shadow on that wall. And so that's more or less how forward scatter is working. It's looking at how much light is diffracted around a cell. So like this here. Side scatter, oh, and I should say its name, forward scatter, comes from the placement of the detector. So in this case, our light source is here. Forward scatter has the detector directly forward or directly in front of the laser. So that's why it's forward scatter. Side scatter, on the other hand, is the detector is at a 90-degree angle to the light source, so over here. So in this case, we're measuring the refracted and reflected light. There we go. And so the more things inside a cell that exist that bounce light, the more light that's going to refract or reflect, the higher side scatter we're going to have. So this is why it's thought of as a measurement of internal complexity. The more things to bounce light, the more side scatter you're going to have. Now, I'm sure you've seen through all your undergrad immunology courses you've done so far, the forward scatter, side scatter for blood cells and how we pull those out. So in this case down here, we'll have our red blood cells, which have really low forward side scatter because they have almost no structure and they're really tiny. Just to the right, we have our lymphocytes. So these also, because they're a small cell, have a fairly low forward scatter. And they're not very complex, so they have a low side scatter. So they tend to fall in this area. Next, we would have our monocytes. So these are slightly larger, slightly more complex. So we'd have a shift in forward scatter and a shift in side scatter. And then finally, we have our granulocytes. So these are about the same size, but much more granular than a monocyte. And so they have an increase in the side scatter. So forward scatter, side scatter is a really nice tool for pulling apart population differences without fluorescence, if there are differences in your populations. All right, so let's do a little game. So we have here a grape, a raisin, a plum, and a prune. So if we were to imagine that these were our cells going through a flow cytometer and looking at forward scatter and side scatter, take a minute and think where on this plot you think these cells would fall. Okay, so let's start with the grape. So when we think of a grape, it's got a smaller size compared to a plum, right? So it's going to then have a lower forward scatter. Now, if we think of the shape or smoothness of a grape, it has a fairly smooth, homogeneous surface, right? There's not really any dents. It's just kind of like a nice, soft, round fruit. So in this case, we'd probably also have a fairly low side scatter. So on our comparison model, our grape would end up somewhere over there. Low forward scatter, low side scatter. Now, if we then take our raisin, this would also have then a small size. So a small forward scatter. What would the side scatter look like? Well, because it has all the wrinkles, it's going to bounce more light. So it's going to have a higher side scatter, placing it around there. A plum, again, very smooth, kind of like a grape, but bigger. So a higher forward scatter, but still a low side scatter. And then last up, we have a prune. It's bigger than a raisin, but still pretty wrinkly. So we have a high side scatter. So that's just kind of a way of picturing how various types of cells would be recognized on a cytometer if we were to turn them into fruits. Hopefully that was meaningful. Can't see your faces, so I don't know. So after our light scatter detection, the part that is really the most exciting are the lasers. So each instrument is equipped with a range of lasers, anywhere between like 2 and 10. And these are the things that provide the qualitative and quantitative data about your fluorochrome labeled molecules. So tells you which fluorescence is expressed on your cells. This is what excites your fluorescence. So determining which lasers excite which fluorochromes comes back to the excitation plots. So in this case, we have 4 different fluorochromes, and you can see this fluorochrome is excited somewhere around this point, and by here we can't excite it anymore. So if we're using a 488 nanometer laser line, which of these fluorochromes will we be able to excite with it? I'll give you a hint. The laser line should cut somewhere through that excitation range. So if you said 2 and 3, you are correct. Fluorochromes 2 and 3 can be excited by the 488 nanometer laser because their excitation profile is within the range of that laser. So if we look then at this fluorochrome, you can see there's kind of two different peaks. So which of these lasers would you use to excite this fluorochrome? Now, if you pick the one that's at the peak excitation, 405, you are correct. So whenever you're looking at which laser you're going to use, you always want to pick the laser closest to the peak excitation point. This will give you the best excitation of that fluorochrome. So downstream of this, we have our optical filters. So once our fluorochromes are excited, we have light that is produced, and we need to have some way of detecting this light, but also seeing what wavelength that light is. So this control of specificity of detection comes from the optical filters. They only allow a set range of light to go through to the detectors. So this is how you're going to determine which light is being emitted from your fluorochromes. Now there's three main flavors of filters. So the first up is a long pass, where any wavelength longer than the one noted will go through the filter, sorry. So for example, this is in the way again. If we have a long pass 500, this will allow anything longer, any wavelength longer than 500 nanometers to pass through. Anything shorter will be rejected by the filter. A short pass is the exact opposite. So anything shorter than that wavelength will go through the filter, anything longer will not. So on a short pass 500, anything shorter than 500 goes through, anything larger does not. And finally, we have the band pass filter. So this is a more specific filter because it only allows a certain range of light to pass through. So if we have a band pass 500/30, that means the center of that band is 500 and it has a total span of 30. So 15 on either side of 500. That means that this filter will let through light from 485 nanometers to 515 nanometers. Any light outside of that range will not go through. So this is how a cytometer chooses which light is going to be passed on to a detector. So if we take a look at these filters, I want you to go through and see if you can determine what range of wavelength would be present on these filters. So if we go through the first one together, we have a 450/50. That means our center is 450 and the total range is 50. So we have 25 on either side. So that means our wavelength range is 475 to 425. Woohoo! So go through and see if you can figure out the last three, and I'll throw up the answers shortly. All right, so there are your answers. So to go through quickly, a 540/30, that means we have a span of 15 on either side. So that gives us a 525 to 555. Again, a 586/16 is a span of 8. So that'll bring us to a 578 to a 594, and a 730/40, span of 20 on either side, so a 710 to a 750. So inside, this thing, man. Inside a cytometer, you'll then have some sort of apparatus that allows the light to pass from filter to filter to filter, ideally without a loss of signal between the filters. So this is a base setup of one of our filters for an instrument in the flow cytometer. Again, this is going to be different for individual machines and how different manufacturers set it up. So this is not a hard and fast rule, but the general theory applies across the board. So in this case, our initial laser light will come in and hit the 735 long pass filter. So that means any light longer than 735 nanometers will pass through. This will hit the 780/60 filter, which will allow in a range of light that's centered around 780, 30 on either side. Okay, so from 750 to 810. And this is the only light that is passed to this detector, which allows us to determine the signal for PE size 7. The remaining light is bounced to the next filter. So it's a 655 long pass. So you'll notice that the wavelength ranges get shorter as we go through the wheel. So this again will allow you to detect a different set of fluorochromes. And then this light continues to bounce through from detector to detector, passing through when it can or bouncing when it can't, until we're able to capture all the light signals that could be collected from our setup on that machine. So this is generally the available fluorochromes for our setup that can be detected. So this is how we're able to get multiple fluorochromes detected from a single laser line. And it's by using filters to break down the emitted light into different pieces. So after all of this, the signals are then passed through onto something called a photomultiplier tube. So in this case, the photons enter and they get multiplied as they go through. This signal is an electronic signal the instrument can understand, and that's how we see our data. We're not going to get into the physics of this today. So giving you one more overview of a cytometer. So you have your fluidic system that brings your cells into the machine, bringing them focused for the lasers and a single cell at a time. In the flow cell, these are where they intersect with your various lasers. The light from these lasers are then sent through an array of filters, which break the light into different parts so that we can identify different fluorochromes. These different light bands are sent onto our photomultiplier tubes, and these are then converted into electronic signals that we can see on our computer and use that to analyze our data to determine which fluorochromes are expressed on which cells. So it's kind of an exciting time to be in flow cytometry because we're in the midst of a parameter space race, really trying to figure out the maximum possible things we can look at in a single cell. And this is a major focus in the field right now, is pushing the number of analyzable parameters in a single cell as high as possible. So when we think of standard flow cytometry, we have a really high throughput technique, but for the most part, our measurable parameters hover somewhere in that like 20 to 30 range. Now this is changing a little bit. So we have some cutting edge new cytometers that have 50 possible channels and are equipped with 6 to 10 lasers. However, even though they have a lot of channels, the current maximum panel size is about 25, and these instruments are extremely expensive just because of the sheer number of lasers that are present. So recently in the field, we've created or moved towards something called spectral cytometry. So this is very similar to a flow cytometer, but differs in how we look at the fluorescence emission. So in our standard cytometers that I've talked about, we have one photomultiplier tube that's grabbing one color. So the one color that comes through that filter. So to add more colors, we add more lasers, more filters, and more PMTs. In spectral cytometry, there are 32 or 48, depending on the manufacturer, detectors that set up to measure the entire spectra of each fluorochrome. So instead of grabbing the band of light that comes through a filter, we look at the entire emission spectra. And so because of this, we can start combining things together that in a single band pass wouldn't work because they'd look the same to the machine. But when the spectra are different enough, we can start using them together. So this has really opened up the number of parameters that you can put on a single cell, the number of fluorochromes you can put on a single cell. And a recent panel that was just published last year had 40 different fluorescence markers on a cell that they looked at on a cytometer with 3 lasers. So that's incredible. So after this, we have something called mass cytometry. So a big thing that limits the number of fluorochromes you can use is the overlap of the emission spectra. So mass cytometry gets away from this problem by using antibodies labeled with elemental isotopes, labeled with metals, because their spectra, for lack of a better word right now, are very discrete. So you can see they have very small peaks as opposed to emission spectra for fluorochromes, which are quite large. You have less overlap in the signals. They're easier to tell apart one from the other. So a mass cytometer, because it's looking at metals, functions a little bit differently than a standard flow cytometer. So the starting point is very similar. We take our cells, we label them with our antibodies, but in this case, the antibodies have the metal tag on them rather than a fluorochrome. These cells are then taken into a mass cytometer where they're nebulized, so they're atomized into nothingness. And the metal isotopes from those antibodies go through a mass spec process. So that's where the mass comes from. It's like a mass spec combined with a cytometer. And so then from each cell, we can look at which metal isotopes are present. And because we know which isotope was on which antibody, we can start to break down what each cell was expressing, similar to how we use fluorescence on a flow cytometer. So here we have a comparison between standard flow cytometry and mass cytometry. And when we look at the profiles between the two, you can see that mass cytometry versus flow cytometry, you get very similar signals detected with the two techniques, even though they're so different. So this shows that we can get or recapitulate the same data using this mass system. Now, these guys then went on to identify all the PBMC subsets using 21 phenotypic markers plus 18 functional markers. So because we don't have that channel overlap, we can have a lot more antibodies added into a single marker. So because of this, in this experiment, they were able to identify all PBMC subsets, plus look at the signaling responses following various modes of activation in a single tube. So this is extremely powerful because you no longer have to break up your sample and look at, in sample 1, I'm going to look at my stats. In sample 2, I'm going to look at my MAP kinases. And in sample 3, I'm going to look at my phospho-ERKs. You can now do that all in a single sample and look at how those changes in activation work together, essentially. So you can study these activation signatures in bulk after various stimuli in a single sample. It's extremely powerful. Taking this one step further even to make it even more exciting, we have something called imaging mass cytometry. So everything I've talked about so far relies on dissociating tissues into single cells, which will remove any potential to study cell interactions or tissue architecture. So mass cytometry really overcomes this and gives you the ability to visualize 37 plus protein markers in the spatial context of a tissue microenvironment. So in this case, you have your tissue section that you label with the metal antibodies. This is then UV laser ablated, put into the mass cytometer, and then after signal extraction and digital processing, you can then start to rebuild these tissues using images. Now this, you might think that, yeah, I can do this by histology, but when you think about it, if you want to do 37 markers by histology, you're essentially taking a slice, do 3 markers, take another slice, do 3 markers. And so by the time you get to markers 37, you're like 13 slices through your tissue. You're no longer looking at the same cells. So in this case, you can look at all 37 markers on a single plane of that tissue, which is just mind-blowing. So this is, again, a comparison between standard immunofluorescence and CyTOF imaging. And you can see that comparing the two, you actually get really amazing tissue architecture, even though this has been laser ablated and then atomized. Like these cells no longer exist, but we can still get a really incredible picture of this tissue. And then you can start looking at all the various markers that are expressed. Now, the one downfall is the human brain can really only interpret 3 signals at a time. So even though you might have 37 antibodies on there, you're still looking at only at 3 in an image. However, when you start looking at the different sets of these, they're all still on that same section of tissue. So you can really recombine them any way you see fit. And the part that still totally gets me is how much detail you get in the tissue architecture of these, even though they've been laser ablated and then nebulized into nothingness. So the one downside of mass cytometry is it is a lot slower. So the throughput is just not there compared to standard flow cytometry. Like while a standard flow cytometry experiment you can usually accomplish in the order of minutes to hours, a mass cytometry experiment of the same size would be more like days. So it is significantly slower. So next up, we have genomic cytometry. So we talked a little bit about this at the start, but I thought I'd loop back and bring it back in here. So genomic cytometry is really the world of single cell sequencing. So in our facility, we run the 10x Genomics instrument. So I thought I'd just give a little overview of how that works. So we have a series of barcoded beads. So each of these barcodes then will be used to identify a single cell. So the barcode is specific to a cell, and eventually you only have one cell associated with a bead. So as these cells get combined into a droplet with a bead, we then lyse the cells and the RNA is released inside still that capsule. So as this gets transcribed into cDNA, we then have the barcode added, which is why all these are different colors, right? So these ones are barcoded with the red signal, these ones with the green signal, these ones with the blue signal. So that's how eventually we're going to be able to go back and determine which transcripts came from which cells. So we then pool this cDNA, and then we can go do transcriptional profiling of these cells. So we know that in cell 1, we have X number of copies of gene 1, X number of gene 2, and X number of gene whatever number. And in cell 10,000, we have these copies. So we can look cell by cell what's expressed, but we can also look on the population and say, okay, well, 10% of my cells express gene 1 and 1% expresses gene 2000. So you get that really detailed knowledge of both your expression profiles on a single cell level, but also how that relates to a population level. And finally, we have a new technique called CytSeq, also sometimes goes by AbSeq. It's got a lot of different names because it's so new. But in this case, it's the combination of single cell gene expression with protein detection. And this is done by using a polyadenylated oligo that is attached, a DNA barcoded polyadenylated oligo attached to the antibody. So instead of using a fluorochrome or a metal isotope, we now can attach an antibody barcode that is genomic that will uniquely identify that specific antibody. And so this oligo tag will mimic a transcript. So when we go and make our cDNA, we will then have transcripts not only for the RNA in the cell, but also for all of the various antibodies that we added. Now, very excitingly, these are unlimited, right? So fluorescents were limited in the number we can add based on the lasers, the filters, and the spectral overlap. In mass cytometry, we reduce some of that spectral overlap, but we're still limited based on the number of metals that we have available to use. In this case, the only thing that limits it is the combinations that you can make inside that antibody barcode. And it's usually about 30 nucleic acids long. So the number of combinations you make are bajillions, to be scientific here. So really the thing that is going to limit this is strictly the cost and how much antibody you're willing to pay for, because each of these have a significant price tag associated with them. So what does this all mean? Well, there's an ever-growing number of techniques in the world of cytometry, and this is allowing you to gain greater and greater insight into your cells and expand your knowledge on a single cell level. It's also just a really fun time to be a cell detective. Cytometry is a very interesting field. It's always changing, it's always evolving, and it's never dull. So hopefully that has gotten you sufficiently excited about flow cytometry. If you have any questions, please feel free to reach out. I'd love to have a chat. Hopefully next year I can see you in person. And on that note, have a great day.